Good afternoon, students. Till now, we have studied various extracellular enzymes produced by microorganisms and how to detect whether the organism produces a particular extracellular enzyme or not. So, we have studied demonstration of enzyme amylase, enzyme gelatinase, enzyme lipase, and today we are going to discuss about demonstration of enzyme catalase. In this experiment, we detect whether the organism produces enzyme catalase or not. So the organism has catalase producing ability or not is to be checked. Now, what is the role of enzyme catalase? See, the catalase test is used to detect the production, presence or detect the presence or production of the enzyme catalase in bacteria. What is the purpose of the test? The purpose of doing the test is identification of gram positive organism, gram negative organism. This is also a primary test used to differentiate between staphylococci and streptococci. Okay, all of you know by now staphylococci and streptococci, right? Staphylococci, if you look at the morphology of the organism appear in bunches, and streptococci are the cocci arranged in chains. So it's one of the important tests used to differentiate between staphylococci and streptococci. Streptococci are catalase negative. Staphylococci are catalase positive. It is also valuable in differentiating various aerobic, obligate, anaerobic bacteria. Okay. So whether the organism produces enzyme catalase or not can be detected by this test. So the test demonstrates the presence of catalase. It is also an extracellular enzyme. So the test demonstrates the presence of catalase, an enzyme that catalyzes the release of oxygen from hydrogen peroxide, meaning that the substrate for the enzyme catalase is hydrogen peroxide. Enzyme catalase breaks the toxic compound hydrogen peroxide and converts it into non-toxic product, that is oxygen and water. Okay, so what is the function of catalase enzyme? The function of catalase enzyme is to break down hydrogen peroxide into water and oxygen gas is released. Okay, hydrogen peroxide is one of the toxic end products that is formed during various metabolic reactions or metabolic activities, and to remove this toxic end product H2O2, hydrogen peroxide, most of the microorganisms except a few genera, most of the microorganisms produce enzyme catalase. The enzyme catalase will degrade the toxic end product of various metabolic reactions, that is H2O2, and convert it into non-toxic products, that is water and oxygen. Okay, so the test is used to differentiate those bacteria that produce enzyme catalase like staphylococci and non-catalase producing bacteria like streptococci. For this test, normally we use 3% H2O2. Okay, but if you are detecting catalase in anaerobic bacteria, actually most of the aerobic and facultatively anaerobic bacteria produce catalase enzyme. But if you are detecting catalase enzyme in anaerobes, you have to use for 15% H2O2. But normally for detecting catalase enzyme, we use 3% H2O2 solution. The bacteria which contain this enzyme catalase are usually aerobic, that they need oxygen, or they may be facultatively anaerobic. What is the principle of the test? The enzyme catalase mediates the breakdown of hydrogen peroxide into oxygen and water. The presence of the enzyme in bacterial isolate is evident when, okay, see, once again, understand that catalase enzyme is responsible for breakdown of hydrogen peroxide into water and oxygen. Whether the organism produces this enzyme or not, that means the test organism, the bacterial isolate given to you, produces enzyme catalase or not, can be checked when a small amount of inoculum is introduced into hydrogen peroxide and there is rapid evolution of oxygen bubbles. Okay, so if you are given a test organism or a test isolate and you are asked whether the organism produces catalase enzyme or not, what we will do? Small amount of culture is immersed or is allowed to interact with hydrogen peroxide, and if there is rapid evolution of gas bubbles, there is effervescence, we are sure that organism is catalase producing, organism is catalase positive. The lack of catalase enzyme, if the organism is not able to produce catalase enzyme, there will be no bubble formation or very weak bubble formation. Okay, but we have to make sure that the culture is 24 hours old. Okay, bacteria actually, by producing enzyme catalase, protect themselves from the lethal effect. Lethal means toxic. So bacteria, by producing catalase enzyme, protect themselves from the lethal effect of hydrogen peroxide, which is accumulated as end product of, okay, aerobic carbohydrate metabolism. As I told you earlier only, that it is one of the end product of various metabolic pathways, and it is toxic. So to protect itself from the toxic effect of H2O2, organisms produce catalase, most of them except a few genera. Okay, so this is the reaction. H2O2, by the action of catalase enzyme produced by microorganism, is broken down into H2O and oxygen gas bubbles. Okay. What is the procedure? If you are doing it on a slide, place a small amount of bacterial culture or colony that is 18 to 24 hours old on a clean glass slide and add few drops, like say 2 to 3 drops of 3% hydrogen peroxide. Okay, and if there is rapid evolution of gas bubbles, effervescence, organism is catalase positive producing, and if there is no effervescence, no rapid evolution of gas bubbles, organism is catalase negative, it does not produce catalase enzyme, right? Tube method, which we generally follow, what we do is we take say 2 to 3 ml of hydrogen peroxide in a test tube, 3% H2O2. Okay, we take 2 to 3 ml of 3% hydrogen peroxide in a test tube, take a small amount of culture on the loop. Care should be taken that the loop should not be made up of iron. Okay, or we can take small amount of culture on a glass rod and immerse the loop with culture or glass rod with culture inside the H2O2 taken in the test tube, and observe for immediate bubbling, immediate effervescence. If there is rapid bubble formation, organism is catalase producing. If there is no bubble formation, no evolution of gas, organism is catalase negative. As you can see from the diagram, the first half is a slide test. This is slide test. You can see we have taken small amount of culture and added H2O2, and there is rapid evolution of gas bubbles. So this is catalase. This organism is catalase positive. And in the lower part, you can see there is no evolution of gas bubbles. When the culture is mixed with 3% H2O2, that means it is catalase negative. Now we can see in the test tube, in the first test tube, you can see there is no rapid evolution of gas bubbles when the culture is immersed in 3% H2O2 in a test tube. So organism is catalase negative. In the second test tube, we can see there is rapid evolution of gas bubbles when the organism is immersed in 3% H2O2. So organism is catalase positive. Is that clear? This is how we can see whether the organism, it's a very simple test to check whether organism produces catalase enzyme or not. Or else what you can do is take 3% H2O2 in a test tube, say 2 to 3 ml of 3% H2O2, and take a nichrome wire loop with the culture and dip it in the 3% H2O2 solution, and you can see effervescence or evolution of gas bubbles. As you can see in this picture, in this diagram, there is evolution or effervescence or evolution of gas bubbles. It indicates that organism is catalase positive, or it produces the enzyme catalase. Okay, so it's a very simple test by which we can determine whether organism is catalase producing or not. So that is all about catalase production by microorganisms.